Welcome to the C45 Association's official YouTube channel. The following presentation is brought to you with support from Sigma Analytical Services with the C45 Association. Sigma Analytical is the only cannabis lab with cross-continental operations in the world and the first cannabis-focused GMP certified lab in Canada. Our service goes beyond a full panel of testing and also covers stability studies, cleaning validation, formulation and product development support, and more. We have been a front runner in testing Cannabis 2.0 products and have validated methods for vape cartridges and a variety of edibles, topicals, and beverages. Sigma is also Shoppers Drug Mart's partner in medical cannabis by Shoppers and Ecolab's exclusive partner in Canada and South America to run cleaning validation studies for cannabis processors and producers. Our short and dependable turnaround times, in addition to accurate and reliable results and outstanding client support, has made Sigma the preferred lab for many LPs. Sigma also offers support on consulting projects for the producers and labs in Canada, Colombia, the US, and beyond. From method development, validation, and transfer, to support for designing, setting up, and running in-house or independent cannabis labs. For any questions or to start a discussion on how we can assist you, please don't hesitate to contact us. We hope you enjoy the following presentation, and don't forget to like, comment, and subscribe to the channel for more quality content.
Welcome everybody to the Sanitation and Cleaning and EM Testing Trending Setting Up for Success, which is not really a full sentence, and I'll get in trouble for even pointing that out to anybody. We're going to have a look at validating your sanitation and cleaning programs, products, testing your water, looking at the different things that are going to affect the sanitation, and ultimately, for those of you that work globally, some of your certifications around GMP, GPP, and other European-based standards. We have three incredibly smart people with us today. We've got Troy Henderson from Virox, Sherry Lawson from Keystone Labs, and Susan Youngquist from Ecolab. I'm going to let them introduce themselves as we get started here, but I'm also going to hit them, as all of my panelists are aware, they never really know what's coming. So we're going to hit them with an intro as well as a first question, and just sort of start this conversation around where we want to go for the next 45 or 50 minutes. As you do your introduction, can you tell us, we'll start at what the importance of having an environmental monitoring program is, because in my world, and again, I do site attestations for Health Canada, and I've been in many facilities. When we say environmental monitoring, I think back to my risk realm, where that actually includes not just monitoring, but you build the facility to be monitored. So how important is that to an organization and to a facility in their business planning and such? So recognizing that very broad question to get us started, we're going to start with Troy Henderson from Virox. So Troy, if you could tell us a bit about yourself and dive in.
Yeah, thanks, Barry. Happy to be here. So yeah, my name's Troy. I work at Virox Technologies. We're the makers of Shield disinfectants. So part of my role at the company is helping implement the product and a lot of the support side of it. So planning trials, protocol recommendations, SOP writing, all of the stuff that kind of goes on behind the scenes on implementing a new disinfectant in your facility. And I guess to answer that question on the importance on EM, it's really important. It can catch potential outbreak in its tracks. It's also good for QA for before and after, if you're doing swabbing in a cleaning scenario or disinfecting scenario, where you want to see if the team's actually doing their job or if they're missing spots or cutting corners. It's really good to have that data and to look at it over time.
Excellent, thanks, Troy. We're going to move over to Sherry Lawson from Keystone Labs. Sherry.
Thank you. I am a quality assurance associate at Keystone Labs, and I was actually a founding member of the company, so I'm very familiar with the work that we do here. We started out as a pharmaceutical company, and now that the world of cannabis is opening up for testing, we're doing a lot of contract testing for cannabis clients as well. A big part of what we're starting to see come in from a lot more clients is EM samples from their production facilities. And I absolutely think that It's critical to have environmental monitoring process and system in place because, as Troy said, the biggest thing is to catch problems before they become a problem. So you want to be seeing your data trending. If it starts to trend into an area of not being in control, you can take action before it actually impacts your product.
Nice. And over to Susan Youngquist at Ecolab, who is touching a whole bunch of our shared clients, but I'm very interested to hear your feedback on that, Susan.
Yeah, thank you very much. So I am a technical consultant and validation manager for Ecolab. Ecolab provides really total plant solutions. So we have cleaning chemistry and disinfection solutions for both environmental spaces and processing equipment. I specifically work with cannabis producers in North America to help with customized cleaning solutions for processing equipment. So environmental monitoring is really important to capture early if there's contamination of some sort or problem that might be starting to develop. And I think it can even extend beyond monitoring the environment, but we can also monitor the cleanliness and quality of our cleaning of the processing equipment as well. So it really reaches beyond just the facility, but into the equipment.
Very nice. Excellent. And that's a great segue. Thank you, Susan, for that, because I'd like to, I know that context is everything and every facility is different, but the goals we're trying to achieve are common. So what are some of the, I guess a better way to phrase this is how would a cannabis producer, how should a cannabis producer be setting up their facility? And I guess we can break this out operationally into two areas. How do they physically set it up? What are some of the key features to the facility? And then further to that, what are they looking to do from an SOPs or an operational point of view to support that facility to ensure that they're not just meeting standards, because that I feel like is a low bar. How are they actually doing it right so that the end product is quality? And we're going to go backwards on this one. So Susan, I'm going to throw you under the bus and start with you on this.
Well, I think the key starting point is to really understand the processes and understand where risk can be present in those processes. I think most participants in the summit are focused on Cannabis 1.0 products, really looking at your growing and cultivation. And I think I'd like to expand the discussion even into those Cannabis 2.0 spaces where you can have different soils introduced because you have different types of products involved. Instead of just the plant materials, you're really getting into the extraction and distillation type activities. You might be working with oil and resin blends, maybe even getting into edibles and topicals, where you're introducing ingredients with really unique characteristics that the traditional cannabis producer isn't used to. So then when we think about process design and even facility design, it's really controlling where these ingredients and potential sources of contamination land within your facility, controlling the flow, people flow, product flow, to try and minimize any kind of cross-contamination.
Is there, before we move on, if I'm a business moving to 2.0 and I'm at the cultivation step, I would hope that the business strategy and business plan recognizes that it's not a pivot without some support. Is there a difference in how you look at that? I mean, you sort of touched on that a little bit, but is there a difference in the facility side? Or is it just in the product?
Well, no, there's a difference even in the facility side. Again, I go back to product flow, people flow, and contamination control. In your growing and cultivation space, day after day, you're working with the same plants, right? The same materials, the same micro-contamination. And then when we start to get into some of these 2.0 products, you can have different actives. Maybe you have different cannabinoids in your different products. so you really want to be careful that you're not somehow cross-contaminating, say, an edible that's supposed to have CBD and not so much THC in it. We don't want that THC getting in there, right? So we really have to be careful with tracking ingredients and material flow through the facility.
Yeah, I can see that. So Sherry, on your guys' side, again, to the broader question, how would you recommend they set up to ensure that, and how does it help you deliver what, I hate to put it this way, but what the client is looking for out of testing, because that has a bad connotation that I'm not trying to infer, but how do you set the clients up for success? What's the advice that you give them?
Well, I would say instead of saying what the client is looking for, what we really focus on is trying to make sure that the results that we are providing to our clients are meaningful for our clients. So instead of just giving them a number, we want to make sure they understand what that number means. Because I think a lot of times, especially coming from a pharma background, we have a lot of expectation to see 0 microbial counts in a lot of our results. And that's an absolutely unrealistic expectation for a cannabis facility. As you're talking about these 2.0 facilities versus the cultivators, you're likely to get fewer microbial counts in the 2.0 because you're not growing plants and you're not dealing with soil and all of those kinds of things. So as Susan said, it's more about managing risk than what the bottom line number is. So I would suggest usually setting up your environmental monitoring program to think about where potential contamination could be coming from. So if you think, oh, here's an air exhaust fan that could be giving potentially impure air or some other kind of impurity into your system, that would be a really good place to have a monitoring site. So we like to try to work with the clients to sort of say, you know, these are the types of things you are looking for. Think about where risk exists and try to mitigate that risk by setting up your environmental monitoring program appropriately to catch that risk and to catch those potential points of contamination before they do become a problem, because that's really the essence of an environmental monitoring, a water monitoring, a cleaning validation. Any of those kinds of programs are set up to mitigate your risk. And the important thing is to catch the problem before it becomes a problem.
Absolutely. It has been interesting to me how often, especially in Western Canada, how many sites we have that are outside of an urban center. So they're using well water, or they're using water that has more dynamics to it, if you will. And they're not concerned about it. And again, well beyond my science background, but that's a huge flag for me. Troy, as you're hearing this, what would you recommend? Again, anything to add to that? And one of the things I would ask you to comment on, what I'm hearing, is that the idea of an EM program gets more and more complicated if somebody is transitioning, or not transitioning, but operating in more than one space. To Sherry's point, if you're cultivation, but you're also production or processing, you really have two different facilities, really, to a certain extent.
Yeah, I 100% agree on that. Kind of as Susan was mentioning, way different soils when you look at processing compared to cultivation. Your protocol should be different based on that. You should have a completely different program. I kind of agree with everything said. More maybe to add on to the physical side of things, in order to make things easier, just making surfaces accessible. Where, and if possible, if you have the ability, like a lot of maybe older facilities might have, exposed piping on their ceilings inside their grow rooms. And that can be an issue when, even getting up there to clean, disinfect, you can foam or fog or, do what you can. But, even then validating that could be really tough or maybe even impossible. So, trying to exclude those areas from your room, any crevices or whatnot, if you have the ability, if you're at a stage where you're adding on, that'll really help and go a long way.
Thank you. That's a great segue for where I wanted to go. Because one of the things, again, as somebody who's not working in the certainly in the QA realm, is just advising to push people to be aware and work with professionals. What are the fundamentals for cleaning? We'll stick with processing equipment at this point. And more importantly, how do you measure? How do you know it's effective? How does that? And I don't know if this changes to the EM program itself, but let's start with you, Susan, just to help me understand a bit about the basic principles, but almost more importantly, how do you validate that it's actually working, happening?
Yeah, good question. And I think it's might be an area that has some new factors for our processors as they move into 2.0. Generally, there's 4 factors that we look at when we're cleaning processing equipment, and we kind of refer to it as TACT, just a way to remember the factors: temperature, action, chemistry, and time. And each unique ingredient chemistry has a different combination of those factors to be able to clean effectively. So like some soils require a really high alkaline cleaner, others might require a high or low temperature to be soluble, and some other soils might be more easily cleaned if you have a high surfactant load. So we do some work with manufacturers up front to figure out how to best clean their soils. Now, that brings the question, what's clean enough, right? Or how do we validate that something's been cleaned? And usually the place I start is visually clean. Can you see any soil remaining on the equipment? And I think that even applies for your environmental space, right? Have you cleaned your walls and your floors and your tables to a point that there is no visible soil? Best in class manufacturers will have visual standards that they use. So actually take pictures so that folks can see what a clean enough surface looks like. I've also seen some do like a white glove test. So equipment might look visually clean, but when you take a white glove across it, there's some residue that comes off. Once you've assured that the equipment's visually clean, then we look at residues of actives, maybe detergent residues, and micro bioburden. And those limits can be based on toxicity or on a therapeutic level of particularly an active. So through some calculations, we can figure out how much residue would be allowed to be left on that surface. And then you can test the surface through either swab testing or maybe collecting a rinse sample and have that analyzed to see how you measure up against your success criteria. As I will mention, it's really important with that kind of testing that you have an experienced and qualified analytical lab working with you. Some of the tests to detect those actives, as a lot of our processors know, and sometimes even the detergents, can be pretty sophisticated test methods.
One of the consistents I'm hearing, and one of the reasons why I've got involved with C45 is that we seem to have such a lack of, this may be too heavy a statement, but a lack of standards around who people are allowed to use and what labs they can pick and choose as compared to going to the best labs. Gets very frustrating in the industry. Sherry, over to you on that. I mean, how does that look coming in your door? Like, how would you support or recommend somebody's working around those fundamentals and validating?
Well, I'll talk about it from an environmental monitoring point of view. And when we're talking about that in a facility context, what we're looking at is either microbial growth or total particulate growth. So these are ways of sampling your air and your environment and your surfaces specifically. So there's 3 different types of monitoring that we would do in a facility. And you would do these extensively during a validation. And then you would, based on your validation data, you would pull back to the more critical sites that you would do on an ongoing basis, just to make sure you have continued control and you're continuing to monitor the microbial state of your production facility. So there's the surface monitoring plates, which just basically has a little bit of agar on the plate, and it's built in such a way that you can actually touch it to the surface. So anything that is on the surface will then be plated onto your plate, and then you incubate it and check it for microbial growth. Active sampling actually pulls a defined amount of air through the sampling device onto an agar plate. And again, you'll be incubating and checking for microbial growth. And then the third way is passive monitoring. So basically you just open a plate to the air and you leave it for an extended period of time while certain activities are going around the plate. So what you're looking for here is routine activities, and you'll get a good idea of how much microbial load is generated through that activity. And as I said, you would set up your sites quite broadly at the beginning for the initial validation, and then based on the validation data, you would be able to pinpoint the most critical points that would give you the best indication that you're starting to trend out of control. And then that's what you would look at kind of on an ongoing basis.
Excellent. Troy, I'm going to throw an add-on into this because as Sherry's talking, and again, mine is a risk background. How do you look at those principles and the validation? And then is it an add-on to the EM program to be responsive? Because we're talking about when you start to see a trend you need to respond to. How do you measure? I'm not sure if it's easy to answer without a specific example, but how do you ensure you're in a position to respond to it? You can have the data, and we talked earlier about knowing what you're looking at, having results being meaningful. But what does that look like for the response side as well as, after you put your fundamentals and your validation?
Yeah, well, I think you need to find the source of the problem or the cause, whatever is causing those numbers to jump. Maybe if you have a microbial load that's much higher than expected, maybe you're not reaching that contact time for your disinfectant, or maybe it was diluted incorrectly. Those would probably be the first two places I would check. But yeah, that's something else I kind of want to talk about too is, as a, processor, you want to clean, and that's really important as well, just as everyone said. But then, disinfection is that next step that you want to achieve. So it's important that, you're not only cleaning, you are disinfecting as well. And, some disinfectants you need to actually pre-clean before you do start disinfecting, and others you might maybe have to do a little bit less cleaning. So make sure that, you're following your disinfectant protocols and your cleaning manufacturer's recommendations.
Is there a danger going backwards? And full disclosure, I can't grow a dandelion. If you're in a cultivation facility, is there ever a danger for somebody that may not be properly trained or may make a mistake or whatever in over cleaning? And that, I guess you should catch that in validation, shouldn't you, before in between crops? I don't know. Susan, is that something you'd see in facilities that is perhaps a slip in training, that you could actually over clean and cause damage to the next round?
Yeah, I think that's possible. I think your validation ensures that your processes are set up appropriately, but your validation doesn't guarantee that every person is always trained and following those procedures. So there's definitely monitoring and tracking and executing the right procedures every time. And overdoing it can cause damage at times. If your cleaning chemistry or your disinfection chemistry is supposed to be used at a particular concentration, more isn't always better. And it could lead to an unsafe situation for an operator, or possibly even cause damage to equipment or, my goodness, to the plants.
I have to assume that we wouldn't see very many of our producers out there or cultivators doing testing or sending testing to a lab in the cleaning phase. Do you guys see that very much? I don't know, Susan and Sherry?
Yeah.
Yeah,
actually we do. That's part of our program is send us samples of your soils, let us do cleaning testing so we can figure out the best cleaning procedure for you, and we can provide those 4 factors that I mentioned to be able to set the best cleaning and disinfection procedure.
And we certainly do see before and after testing. So somebody will have us monitor some plates prior to the cleaning procedure being done, and then they can do some targeted cleaning and see what the difference is once the cleaning has been completed. And it sort of gives a good idea of whether or not the cleaning protocols they have in place are effective or not.
This very much speaks to having somebody that knows what they're talking about. Go ahead, Troy.
Yeah, I just wanted to add in there, echoing which everyone said, it's really good, if you have, let's say you're doing ATP, if you're monitoring using that, and you have your limit, whatever it's set at. But it's also, we've seen a lot of value is doing your before and after and seeing, what you reduced it by. If you started at 23,000 and got down to 400, that's really good. But if you started at 500 and got down to 400, maybe that process wasn't followed, and you can really see how good of a job was done. So that's what we always recommend. It's not always possible for every producer. It's like an extra swab, which is a little bit more cost. But if you can do that, or even sometimes do that, the data you get is extremely valuable.
Is it possible to generalize the cost of, I mean, on the risk side, we always can build matrices out of what happens if you don't do these things. But it sounds like, as I'm learning from you guys, that from a business strategy, a business planning perspective, these are critical steps that I would be really shocked to hear somebody not doing these kind of things. Is that kind of a fair position? Yeah, everyone's being really careful. Well done, well done. Well, and the reason I put it that way is within the Canadian industry, we're now dealing with so many micros coming into the realm, and there is a fairly significant gap operationally, and certainly in my realm around security requirements and those types of things. And I wonder how many of our micros are coming out the door and within their business planning, not factoring in some of these types of testing and monitoring or a good EM program because they don't feel they're big enough for it. And I know with all things, size has an impact, but maybe we could just speak to, how important is this, or are there pieces or ways to do this that a smaller producer and a smaller processor can still achieve some of those goals without it bankrupting them? If we can, we'll start with Sherry on this one.
Okay. I think you have to think in terms, when you're thinking in terms of scale, the scale is actually important because for a big producer, they're going to have a larger facility. They're going to have larger batch sizes. So having to reprocess a batch because of microbial contamination or having to discard a batch because of, for instance, aflatoxin contamination it's a larger amount of product. But in terms of percentage of their overall worth, I mean, a micro producer has a smaller amount of product overall, but it's still a significant amount of their capital is tied up in that product. So I think absolutely it's an expensive thing to undertake. And it's a little bit harder to justify just because there's no regulation saying you have to test to within these particular numbers when you're looking at your environmental cleaning, all of that kind of thing. So it's easy to say, oh, we don't have to do it because the regulations don't really specify where we need to be, on this scale. But when you think in terms of the danger to your product, I think it's an important risk mitigation that all companies do need to consider. Absolutely, it's a cost up front, but it helps to prevent loss of product long term. And I think it also helps to train the staff to help them understand these are the potential points of contamination. So these are the places that you really need to be careful when you're performing your job function. So having these programs in place gives a little more awareness to those types of risks and hopefully a little bit more care when the work is being done.
We'll go over to you, Troy.
Yeah, no, I completely agree with that. It's difficult for micros because they're starting small, and it could be some of these units can be couple $1,000s. But I know if I was a quality assurance person, I would have trouble sleeping at night not knowing what's going on in those rooms. And once you see that data and once you know what you can be tracking, I think it's a no-brainer. Like if you're a micro, let's say you have $100,000 in a room. It's minuscule compared to that. And for the large producers, you might have millions of dollars in a room. That really, it really makes sense for them. And, for micros, maybe at a bare minimum, just do one surrogate surface in each of your rooms, between your crops. And that's a bare minimum, but that'll at least give you something to work off of. And start there, and maybe you see some more cash open up, and you can afford, to do 2 swabs or to do one swab during production or whatever that is. So that's something that we try to work with them on, and just showing them that the value and and the risk associated with it.
Yeah, I'm really hearing that benchmark and that risk mitigation really is critical, not on a business plan. And Susan, you work with a number of the bigger players on that business plan. I don't want to say dollars don't matter, but they're looking at any losses as being not acceptable losses. So to a micro, it's the same thing, right?
Yeah, and I think it comes down to their reputation, really, and the quality of their product. I think every manufacturer, regardless of size, wants to be known as a quality manufacturer.
Absolutely.
If I can add to that, Barry.
Yeah, go ahead, Troy.
Sorry, if I can add one more thing too for the micro side. Health Canada, they really love data, and having that the EM program and showing them that you're validating that. And there's even other things you can do, like you can, a lot of disinfectant products have test strips that you can validate that are being disinfected at the proper dilution. And you can create a logbook if you have like a dilution station. And we've worked with a lot of facilities setting up all this data collecting. When Health Canada comes in, they're seeing, wow, you're doing everything on the front end and the back end. Like you're really doing everything you can. So maybe if you're not even looking at it as a risk for, the revenue side or the crops, it'll make your audits a lot easier showing Health Canada, assuming your data is good and you don't have crazy numbers, it'll make your audits a lot easier.
That's very.
I have to admit, one of the things I'm despite the bumps we've had over the last many years, that gives me a lot of hope is that we seem to be moving into more business fundamentals around data collection, data validation across the business scope, not just in QA, but we're actually, and some people will be upset that I say this, but we're seeing more business focus and business fundamentals applied now, which is going to help us with the sustainability of the industry. I guess the last question I want to hit you guys with is a really, really broad one, and you can decide how far you want to lean in on this. But can you pin down one or two or three of the most common mistakes made by producers in the applications of their disinfectants and sanitizers? What are they doing that they could change to come up with a better success? We'll start with you, Troy, and then work through the list again.
There's definitely a lot. I would say number one, and really on topic for what we've been talking about, is the failure to properly pre-clean. A lot of people will maybe use isopropyl alcohol. I don't want to pick on a specific chemistry, but maybe use that as a cleaner and disinfectant. And we all know that, it really should be one or the other. And that kind of leads to the second mistake I see, which is the failure to reach the contact times required by the product. So if you have a 10-minute contact time, that surface needs to stay wet for 10 minutes if you want to achieve that. So that's another one. But I think the third one would be, overall, maybe kind of what we were talking about earlier, doing a little bit too much, and not in terms of because they're, maybe concerned and they want to get those low numbers. I just mean like cleaning smart and looking at ways that you can maybe do this a bit easier. There's there's equipment you could purchase that, maybe you don't want to, or you do, that can make it easier. But even just by protocols and listen to the people on the floor, the users, they might have ideas. And if you want to maybe switch protocol, that's always a good idea to do a trial, validate that it will work. Maybe it'll be better or at least the same. Maybe it'll save you time, and work forward like that instead of maintaining status quo and being okay with maybe it taking 3 days to clean a 20,000 square foot room.
Nice. Sherry, over to you. You're going to see some of these coming through your door.
Yeah, I mean, I'm coming at it as a microbial environmental monitoring perspective. And so really the problem would be not doing the testing at all, because there's still, like you said, it's an easy justification to make. I don't have to pay for this. I don't have to show this data. This isn't a requirement. Regulatory bodies are really pushing, although I do expect that it's going to become more and more of an actual regulation rather than just kind of this is a good thing to have. But having said that, we are seeing more and more cannabis customers coming through with EM and wanting to understand what their EM results mean. I mean, obviously, especially molds, that's really toxic and really dangerous because it can introduce the aflatoxins. I mean, when you're looking at bacterial contamination, you can just do irradiation, you can do some reprocessing to mitigate that problem. But once you have specific types of mold in there, you can't get rid of the aflatoxin, and that batch is gone. So we see clients coming through testing for aflatoxins, and really the first piece of that is testing your environment for the presence of bacterial or microbial contamination. So I think the industry is really starting to move in that direction and understanding the importance of an environmental monitoring system. And that's a really fantastic thing to see. But I would say that that's probably one of the biggest mistakes that I would see is companies just not thinking it's worth it up front.
I'm so glad not glad to hear you point it out, but we keep hearing it. And I hope we're moving past the race to the bottom line of just meeting regulatory requirements or lack of drives me absolutely crazy. Susan, you work directly in these environments, so over to you. What are you seeing, and are they fixable? Again, I want to lead a little bit. Sorry, again, I'm throwing you under the bus, adding to this, because some of the big environments you work within, if there's problems, that's a big fix. So, what does that look like?
I think Troy hit a couple of really solid ones with ensuring that the disinfectants are at the right concentration and have the right contact time. And he also mentioned the pre-cleaning importance, and that's one that I think manufacturers really have to keep an eye on. There's a tendency to do that upfront cleaning development and get through cleaning validation, and then just assume that procedure is going to be good thereafter. And we have to continually monitor that our cleaning is effective. Because anytime you get a soil buildup and you start to get microbial growth in that soil layer, you can develop something referred to as like a biofilm that can be very, very difficult to break through with your regular cleaning procedures. And then you get these pop-up contaminations that can go on for months or years, and you can't find the source. So if we can just stay on top of it and ensure we're cleaning and monitor that cleaning and disinfection all the way along, we can maintain that higher quality.
I know it's not apples to apples, but I am amazed at a human level how within science and QA and within my realm of prevention risk and such, how hard it is to sell prevention. Because I'm sure we've all had these discussions with clients, and it's no, I'll wait till it happens. And they just don't understand how big that cost is if you wait till it happens. So excellent information. A quick roundtable. Is there anything we missed along the way here before we roll into the Q&A side of all of this? Okay, hearing nothing. I run meetings this way too. Go ahead, Susan.
I would add one thing. I encourage everybody to reach out to their experts, right? We've got great experts here on this panel, and we're all available for all the manufacturers to reach out to. So use those resources.
Absolutely. All right, I will see all of you, I hope, on the other side of the Q&A here, and we'll see what comes from the crowd. Thank you so much. You've certainly educated me. The dangerous thing is I now have a whole page of notes, so that if they don't ask questions, I will be. So this could be really bad. And I would be remiss if I didn't do a shout out to Jody McDonald's frog hiding behind Sherry. That is an awesome. Oh, there we go. All right, we'll see everybody in a couple of minutes.
Thank you.
Thank you.
Thank you.